Hello, my name's Peter Crane. I'm the president of the Oak Spring Garden Foundation, and I thought I'd just take a few moments to explain a little bit more about the paper that was published in Nature in May of 2021. A characteristic feature of all angiosperms is that the seeds develop inside fruits. So if we look at the developing fruits of a peach or an apple or a citrus, we can see the seeds developing inside. One key question for understanding the evolution of angiosperms is, where did the fruit wall come from? Can we see its origins in any of the close angiosperm relatives in the fossil record? But before coming to the question of the fruit wall, we also have an issue to deal with pertaining to the seeds themselves, because examining the ovules from which the seed develops in angiosperms, flowering plants, we see that the nucellus, the structure in the middle of the seed that encloses the egg cell and later the developing embryo, is surrounded by two tissue layers, the so-called inner integument and an extra outer layer, the outer or second integument. Like the carpel itself, this outer or second integument is a characteristic feature of angiosperms, of flowering plants, because in other seed plants, such as conifers and ginkgo and cycads, there's just one integument, which most people think is equivalent to the inner integument in angiosperms. So we can think of the angiosperm fruit as a series of vessels, one inside the other, like a Russian Matryoshka doll. In the center is the nucellus that contains the egg cell and that will develop into the embryo. This nucellus is enclosed within an inner integument, equivalent to the integument that we see in ginkgo and cycads and conifers. In turn, the nucellus and the inner integument are enclosed within a second integument, the outer integument, characteristic of flowering plants, and the whole structure that will develop into an angiosperm seed is enclosed within the carpel. So the first priority in understanding the origin of angiosperms is to explain where the second outer integument comes from. For over 100 years, we've known that several groups of extinct plants from the Mesozoic, especially the Jurassic plant Caytonia, had their seeds enclosed inside recurved structures referred to as cupules. Originally, it was thought that the cupule might be homologous to the carpal wall, but over the last several decades, it's been thought much more likely that the cupule is actually homologous, fundamentally comparable to the outer second integument of an angiosperm seed. This idea is also attractive because the basic form of the angiosperm ovule is also recurved, like the cupule of Caytonia. And we now know that the genetics controlling the development of the outer integument in angiosperms is linked to that curvature. And also that the developmental genetics of the outer integument is very different from the genetics controlling the development of the inner integument. The importance of the discovery from the Zanahiyr open-cast coal mine at Jarod Bana in eastern Mongolia, China, is twofold. First, it provides a new and very detailed window with superb preservation into the extinct plants of around about 125 million years ago, very close to the early diversification of flowering plants. And second, it provides very detailed information on the form and anatomy of a new kind of recurved cupule, which enables careful comparison with previously described fossils. And those comparisons lead to a unifying explanation for the several kinds of cupules that have been described previously, which have often been regarded as of separate, independent evolutionary origins. Together with other recently described fossils from Mongolia, British Columbia and Virginia, as well as previously described material from Antarctica and South Africa, we can now see that these cupules varied considerably, but all on the same fundamentally similar plan. All are modified axes bearing terminal seeds inside the recurved cupules. In the material from the Zanaher open cast coal mine, the reproductive axis bears just one cupule that has two seeds inside. In this video, you can see 4 seed-bearing axes isolated from the chert by X-ray computed tomography. Each axis bears a cupule, coloured blue, and each cupule contains 2 seeds. You can only see one in the upper cupule here, coloured brown, and each seed-bearing structure is borne in the axil of a bract, coloured green. This is a 3-dimensional printout of the top 3 cupules in the previous video, and as you look at it, you can see the cupule itself, the bract, and also one of the seeds still inside the cupule. Each cupule is about half a centimetre long. Here you see a cupule in longitudinal section. You can see the bract projecting to the left, the cupule enclosing a single seed that is attached near the apex of the cupule. As the video proceeds, you'll see the first seed slowly disappearing, and then the second seed slowly starting to come into focus, also attached at the apex of the cupule. In the recently described material from Mongolia and British Columbia, the reproductive axis is branched, with each branch bearing a single cupule and each cupule having a single seed inside. In recently described material from Virginia, the reproductive axis is even simpler and bears just one cupule that has one seed inside. In other material from Mongolia, the reproductive axis is flattened and 3-lobed, and on the upper surface of each lobe there's a single cupule, each with one seed inside. Catonia is a similarly flattened axis that bears many cupules, each of which contains many seeds. Triassic fossils from Antarctica provide a further variant in which the axis is proliferated, just as it is in Catonia, and bears several cupules, but each cupule contains only one or sometimes 2 seeds. And the variation among all these different kinds of cupules is probably explained by differences in pollination and dispersal in these extinct plants. The implication of all this is that the second outer integument of the Angiosperm seed is likely directly fundamentally comparable to the recurved cupules seen in all of these different extinct Mesozoic plants. So the key question now is, which of these groups is most likely to be most closely related to flowering plants? Answering this question will require much improved information from the fossil record. For example, more detailed investigation of the seed-bearing structures of a critical group of extinct plants from the Triassic of the Southern Hemisphere. But among all of these plants, as others have suggested previously, Catonia is currently the top candidate for an especially close Angiosperm relative. And it will also be interesting to see if this unified explanation can be extended to other enigmatic seed-bearing structures that are preserved in the Mesozoic fossil record.